Reactions of drug metabolism. The kidney cannot efficiently eliminate lipophilic drugs that readily cross cell membranes and are reabsorbed in the distal convoluted tubules. Therefore, lipid-soluble agents must first be metabolized into more polar hydrophilic substances in the liver using two general sets of reactions, called phase 1 and phase 2. Phase 1. Phase 1 reactions convert lipophilic molecules into more polar molecules by introducing or unmasking a polar functional group, such as hydroxyl or amino. Phase 1 metabolism may increase, decrease, or leave unaltered the drug's pharmacologic activity. Types of phase 1 reactions. Phase 1 reactions utilizing the P450 system. Phase 1 reactions not involving the P450 system. Phase 1 reactions utilizing the P450 system. The phase 1 reactions most frequently involved in drug metabolism are catalyzed by the cytochrome P450 system, also called microsomal mixed function oxidases. Oxidation is the addition of oxygen to the substance or the removal of hydrogen from the substance. In electronic terms, loss of electrons is called oxidation. The oxidation proceeds by the drug binding to cytochrome P450. These enzymes unmask or add polar functional groups. This results in a metabolite that is more water-soluble, enabling it to be easily excreted through the kidneys. The P450 system is important for the metabolism of many endogenous compounds such as steroids, lipids, etc., and for the biotransformation of exogenous substances, xenobiotics. Cytochrome P450, designated as CYP, CYP, is a superfamily of heme-containing isozymes that are located in most cells but are primarily found in the liver and GI tract. Nomenclature. The family name is indicated by CYP added to an Arabic number, followed by a capital letter for the subfamily. For example, CYP3A. Another number is added to indicate the specific isozyme, as in CYP3A4. Specificity. Because there are many different genes that encode multiple enzymes, there are likewise many different P450 isoforms. These enzymes have the capacity to modify a large number of structurally diverse substrates. In addition, an individual drug may be a substrate for more than one isozyme. Considerable amounts of CYP3A4 are found in intestinal mucosa, accounting for the first-pass metabolism of drugs such as chlorpromazine and clonazepam. Genetic variability. P450 enzymes exhibit considerable genetic variability among individuals and racial groups. Variations in P450 activity may alter a drug's efficacy and the risk of adverse events. CYP2D6, in particular, has been shown to exhibit genetic polymorphism. CYP2D6 mutations result in very low capacities to metabolize substrates. Some individuals, for example, obtain no benefit from the opioid analgesic codeine because they lack that CYP2D6 enzyme that demethylates and activates the drug. The frequency of this polymorphism is in part racially determined, with a prevalence of 5 to 10% in European Caucasians, as compared to less than 2% of Southeast Asians. Inducers. The CYP450 dependent enzymes are an important target for pharmacokinetic drug interactions. One such interaction is the induction of selected CYP isozymes. Xenobiotics, chemicals not normally produced or expected to be present in the body, may induce the activity of these enzymes by inducing the expression of the genes encoding the enzyme or by stabilizing the enzyme. Certain drugs, for example, phenobarbital, rifampin, and carbamazepine, are capable of increasing the synthesis of one or more CYP isozymes. This results in increased biotransformation of drugs and can lead to significant decreases in plasma concentrations of drugs metabolized by these CYP isozymes with concurrent loss of pharmacologic effect. For example, rifampin, an anti-tuberculosis drug, significantly decreases plasma concentrations of human immunodeficiency virus HIV protease inhibitors, thereby diminishing their ability to suppress HIV variant maturation. This figure lists some of the important inducers for representative CYP isozymes. Consequences of increased drug metabolism include decreased plasma drug concentrations, decreased drug activity if the metabolite is inactive, increased drug activity if the metabolite is active, decreased therapeutic drug effect. Inhibitors. Inhibition of CYP isozyme activity is an important source of drug interactions that lead to serious adverse events. The most common form of inhibition is through competition for the same isozyme. Some drugs, however, are capable of inhibiting reactions for which they are not substrates. For example, ketoconazole, leading to drug interactions. Numerous drugs have been shown to inhibit one or more of the CYP-dependent pathways of warfarin. For example, omeprazole is a potent inhibitor of 3 of the CYP isozymes responsible for warfarin metabolism. If the two drugs are taken together, plasma concentrations of warfarin increase, which leads to greater inhibition of coagulation and risk of hemorrhage and other serious bleeding reactions. Note: the more important CYP inhibitors are erythromycin, ketoconazole, and ritonavir because they each inhibit several CYP isozymes. Natural substances may also inhibit drug metabolism. For instance, because grapefruit and its juice inhibits CYP3A4, drugs such as nifedipine, clarithromycin, and simvastatin, which are metabolized by this system, persist in greater amounts in the systemic circulation, leading to higher blood levels and the potential to increase the drug's therapeutic and/or toxic effects. Phase 1 reactions not involving the P450 system. These include amine oxidation, for example, oxidation of catecholamines or histamine, alcohol dehydrogenation, for example, ethanol oxidation, asteresis, for example, metabolism of pravastatin in liver, and hydrolysis, for example, of protein. Phase 2. This phase consists of conjugation reactions. If the metabolite from phase 1 metabolism is sufficiently polar, it can be excreted by the kidneys. However, many phase 1 metabolites are too lipophilic to be retained in the kidney tubules. A subsequent conjugation reaction with an endogenous substrate such as glucuronic acid, sulfuric acid, acetic acid, or an amino acid results in polar, usually more water-soluble compounds that are most often therapeutically inactive. A notable exception is morphine 6-glucuronide, which is more potent than morphine. Glucuronidation is the most common and the most important conjugation reaction. Neonates are deficient in this conjugating system, making them particularly vulnerable to drugs such as chloramphenicol, which is inactivated by the addition of glucuronide acid, resulting in gray baby syndrome. Note: drugs already possessing an OH, NH2, or COOH group may enter phase 2 directly and become conjugated without prior phase 1 metabolism. The highly polar drug conjugates may then be excreted by the kidney or in bile. Reversal of order of the phases. Not all drugs undergo phase 1 and 2 reactions in that order. For example, isoniazid is first acetylated, a phase 2 reaction, and then hydrolyzed to isonicotinic acid, phase 1 reaction. For studying pharmacological science, go to the link in the description box below this video or in the comments section. Thanks for watching and happy studies.